In this lecture, we are going to cover glycogen metabolism. Metabolism of glycogen involves its anabolism, that means synthesis of glycogen from glucose, and its catabolism, its degradation into glucose molecules. Glucose originates either from diet or from gluconeogenesis. And as energy needs in the cell arise, glycogen is degraded into glucose, which then enters bloodstream and eventually glycolysis. Glycogenesis means synthesis of glycogen. Glycogen is synthesized starting with glucose 6-phosphate. Enzyme phosphoglucomutase catalyzes equilibrium reaction. It equilibrates glucose 6-phosphate and glucose 1-phosphate. Glucose 6-phosphate is substrate for glycolysis, while glucose 1-phosphate is substrate for gluconeogenesis. Therefore, depending on the need of the cell, one of those will be generated and present in larger amount. Glucose 1-phosphate is converted into activated glucose molecule, one that can be added to glycogen chain. And that's done by enzyme inorganic pyrophosphatase. Glucose 1-phosphate reacts with uridine triphosphate to give uridine diphosphate glucose. That reaction actually energetically is not favorable. Equilibrium is about 1. So on its own, equilibrium would be halfway towards the product. But product is inorganic pyrophosphate, which under reaction conditions spontaneously hydrolyzes into 2 phosphate molecules. And that process makes the overall process favorable. And so, because of the hydrolysis of inorganic pyrophosphate reaction byproduct, the overall process is favorable and reaction goes to completion. So glucose 1-phosphate is completely converted to uridine diphosphate glucose. And then uridine diphosphate glucose reacts with existing glycogen chain to add 1 molecule of glucose. Now, to form glycogen, chain cannot be generated on its own. Chain cannot start on its own. Small protein glycogenin first has to be generated, and that's a starting point to which individual molecules of glucose are added and glycogen starts forming. So the overall reaction is glucose 1-phosphate reacts with glycogen and uridine triphosphate and a molecule of water. Molecule of water is actually the one that hydrolyzes pyrophosphate molecule. And then reaction products are glycogen extended by that 1 glycose unit, uridine diphosphate, and 2 molecules of inorganic phosphate. In the opposite process, glycogenolysis, glycogen is phosphorylated at terminal molecules of glucose. The end molecules of glucose are phosphorylated and then they are released as glucose 1-phosphate. Then enzyme phosphoglucomutase that you have already seen just a minute ago as enzyme involved in glycogenesis. Same enzyme isomerizes glucose 1-phosphate into glucose 6-phosphate. Finally, glucose 6-phosphate is hydrolyzed to glucose, which then is released from the cell into the bloodstream. It increases blood levels of glucose, and eventually glucose is taken up by other cells and enters glycolysis to provide energy. Glycogen metabolism is mainly regulated by hormones. And hormones that are involved in glycogen metabolism are glucagon, which activates enzyme glycogen phosphorylase that results in release of glucose into the bloodstream. Glucagon is pancreatic hormone. It's a small peptide and its main catabolic hormone. Another pancreatic hormone, which is also a peptide, polypeptide, is insulin, and it has opposite effect of glucagon. When blood levels of glucose are high, insulin is released and it inhibits enzymes of glycogenolysis and activates enzymes of glucogenesis. Glucagon and insulin have opposite effects and they act together to regulate normal levels, to keep blood levels of glucose normal. Finally, another hormone, and that's adrenal hormone, is also involved in glycogen metabolism. It is released in response to stress, and it activates glycogenolysis and inhibits glycogenesis. This completes this very short lecture on glycogen metabolism.